The skeletal system is the structural framework of the body composed of approximately 200 bones in adults. The bones have an inner trabecular layer composed of cancellous bone and an outer cortical layer composed of compact bone. Bones are composed of 4 types of cells, namely osteoblasts, osteocytes, osteoclasts, and lining cells. The formation of the bone is carried out by active osteoblasts. Osteoblasts synthesize type 1 procollagen molecule into the ECM, later forming collagen matrix. They also synthesize osteocalcine and osteonectin that bind to collagen and to hydroxyapatite crystals, a crystalline mineral of the bone. Calcium is then deposited in the form of calcium phosphate, while hydroxide and bicarbonate are added and the hydroxyapatite crystals are formed. As the bone matrix is formed, osteoblasts cease synthetic activity and become osteocytes embedded in the bone matrix. These osteocytes are interconnected via canaliculi, which allow transfer of calcium ions from the interior of bone to surface, in the process called osteocytic osteolysis. Bone resorption, on the other hand, is carried out by osteoclasts, which are multinucleated cells that resorb calcified bone. Osteoclasts not only extract the calcium from the bones, but also destroy the bone matrix, resulting to a decrease in bone mass. Osteoclasts attach to the bone using integrins that bind to vitronectin found on the surface of the bone matrix. This attachment creates a lacuna between the ruffled border of the osteoclast and the surface of the bone. Osteoclasts contain carbonic anhydrase inside the cell, which provides the cell with high intracellular hydrogen concentration. The high hydrogen concentration is used by the osteoclasts in order to secrete acids and proteases via the V-type hydrogen pump into the lacuna. The carbonic anhydrase also forms bicarbonate inside the cell, which is removed via chloride bicarbonate exchangers, which are found on the membrane opposite the side of lacuna. The acidic environment in the lacuna dissolves the bone mineral, while the acid proteases hydrolyze the matrix proteins, destroying the bone matrix. After resorbing some of the bone, osteoblasts replace osteoclasts in order to promote new bone growth and mineralization on the same site. Parathyroid hormones indirectly stimulate bone resorption by binding to receptors on osteoblasts, which stimulates the release of IL-6 and rank ligand, which promote resorption. The rank ligand binds to the rank receptor on the osteoclasts, stimulating bone resorption. Bone resorption is also stimulated by parathyroid hormone and vitamin D. These 2 factors stimulate osteoblasts to release MCSF, which promote proliferation of osteoclasts. Osteoblasts also have a regulatory mechanism which protects bone from resorption. Osteoblasts secrete osteoprotegerin, which when bound with rank ligand protects the bone from osteoclastic activity. Calcium is a major component of bones. It serves a multitude of functions like the creation and maintenance of action potential, muscular contraction, motility, cytoskeletal rearrangements, cell division, cellular secretion, and in modulation of enzymes.